Hello again, this is Fredrik Wermeling, and this is the part 3, the third video in this playlist where I'm describing how my group is using CRISPR screens or analyzing CRISPR screens. And this is the first video where we're going to start to actually analyze the data sets, right? So in the previous two videos we looked at how to process data either from your own data coming from a GEC or from DepMap. And now we're going to start using a first tool to analyze, and here we're going to use string database, which is a fantastic resource. In the previous video we downloaded data sets from DepMap to look at genes that affected A549 cell line. Here we have then an example of genes that when knocked out positively influence the cells, so you get rid of these and the cells are happy, they relatively expand. And now we're going to analyze and try to see if there's any patterns in here. So I'm going to copy this list, I'm going to go to string, I'm going to remove myself. And here you have search and then multiple proteins. You paste your list of genes, you choose the organism, in this case Homo sapiens, press search. And here comes up, you have a potential here or to sort of change genes. So it shows you several genes that has the name that was searched for in their description. I never seen any reason to change these, but if you have sort of domain specific understanding here, maybe you could look into it. But in general, I always press continue here. I never seen a reason not to. And what you then get out is this type of network. I can just show you can move these around if you want to make them look a little bit more. If you think you can make them look nicer, and you can press the genes and you can get some information about it. You get some structure. You can press the interactions. So here we have interaction between TP53 and CDKN1A, and you see that this had 3 lines here, and the interaction was done based on 3 functional links here, one being experimental data, association and curated databases, and co-mention and PubMed abstracts. And you can press these sort of things to get up sort of more supporting data for this. So again, this is a fantastic resource. It has so much data in it. I think it's really impressive. You can then, I can mention that these images for presentation, I usually just make a screen dump or just a screen grab like this and put it into a snippet tool to look at it. But you can also export it if you want to use it for publication. Of course, please refer to that you're using string database in that sense. So you can export this image as vector or bitmap graphic, for example, or you can also export as the names of these genes or proteins. You can look at enrichment in the networks based on Go terms. There are KEGG pathways, React Home, Wiki pathways, etc. Several of these different ones. And here, if you're interested in sort of regulation of protein stability, if you press it, you can see which genes it is, and you can actually press several of these to sort of see if there are overlaps between them. You see here that there's 6 of the genes here, and again these are the 6 genes that were found in this network, and you can press either of these to sort of make a new interactome with those. You can also press the whole network. Of course, the larger networks become, the more sort of the relevance gets a little bit lost there. But there's a lot of things you can click to sort of identify things here. You can get this data in these tables, and I just want to highlight, you can press more to see more of it. You can get it also as a graphic representation like this. You can change whatever you want to look at here. And these can also be downloaded as both vector and bitmap graphics. So again, this is a fantastic resource, I think, both for making images and to identify sort of pathways that are enriched. And I think it's an important point to make. And again, when you have a lot of genes, it's going to be harder to see this. But what you want to typically see is that we have this sort of networks, that genes are interacting. There are certain interaction between genes. This doesn't mean that this gene is not real or not important in your screen setting, but I would definitely suggest that you start focusing on the enriched or the genes that are connected when you try to understand your hits in your screen. Okay, so now very briefly, let's look at also the other end of these data. So again, the dropouts here, the genes that when you knock them out, the cells are not happy in this particular case, are usually very many, between 1,000 and 2,000. They are often pretty stereotypic in how they are, in the pathways they're involved in, and since this is a larger data set, it's going to take a little bit more time, but with the magic of video editing, I think it's still going to be fast. So I pasted genes, Homo sapiens, search, press continue again. I don't think there's anything wrong here. It's when there's a lot of genes, it's saying basically this is too much, I'm not going to be able to make things looking as fancy, etc., but just press continue. So as you see, and this took roughly a minute, I cut away the waiting part, but as you see, this type of networks doesn't really add too much understanding or that useful. So too many genes, not super relevant. But what you can see here as a final thing then is that, of course, now you're going to start to have some really massive, significant pathways that are identified, and they're going to be fairly stereotypic in their sort of patterns. It's going to be genes that you could imagine are important for cells, RNA, ribosomes, translation, splicing, peptide biosynthesis, mRNA processing, gene expression, tRNA, metabolic processes, etc., etc. So you see there are a lot of these fundamental pathways that are relevant for cells, as you can imagine. That comes up. So this type of dropouts are not necessarily always very relevant to look at. Again, you can look at depth map, and one of the most essential genes that often comes up is RAN, for example. There's many of them, but you can see that the gene effect value here is -4 for CRISPR in general. So it's a very, very strong effect. And again, this is a histogram showing. And depth map defines everything below 1 essentially as a common essential. Right? So it's typically so that there is a set of genes, 1,000 to 2,000, that really are very negatively impacting cells. What becomes interesting to look at in a data set like this is, of course, is there anything in your cell line that is very strongly relevant for the cell, very essential for the cell, but that is not maybe common? Is there something that sticks out there that makes you understand something about the cell or how you can target it for therapies? And that will become a topic for another video. So with that, with this messy interactome, I leave you. This was the third video. I hope you learned something, and there will be more videos where we go into and I show you different ways of analyzing this type of data.